Okay, in yesterday's lesson, we took a look at the structure of DNA and how DNA copies itself through a process called replication. Today, we're going to take that one step further. We're going to take a look at how cells use their DNA to actually make an end product, and that end product in the long run is something called proteins, which you've learned about before. And in order to do that, DNA has to use a close cousin called RNA. So today's little lesson is all about RNA, the structure of it, how it works, and how DNA gets turned into a copy of RNA through the process called transcription. So first things first, overall, here's where we're going over the course of the next couple of lessons. We're starting off with here in this particular part that we looked at yesterday, which was how DNA copies itself through the process of replication. We looked at the structure of DNA, the double helix, looked at the backbone, which is the sugar phosphate molecules, and then we looked at, most importantly, complementary base pairing, because complementary base pairing was how the process of replication was accomplished in a very straightforward, error-free kind of message or process. In the next two lessons, we'll be taking a look at how DNA actually gets used. So we're kind of going this direction now in terms of DNA has to get copied into a functional version of it called RNA, and this is a process called transcription, which is what we're looking at today. And then tomorrow's lesson will focus on the next step when we take that RNA copy and then turn it into a protein, and that process is called translation. So today's lesson, we're really focusing on this part. Yesterday's we looked at this part, today's here, tomorrow's there. So there's the kind of different steps that we're at. So we're looking at the DNA to RNA transcription process through the rest of the lesson today. So, first of all, why do we actually have to use transcription at all? Why not just use the DNA directly to make proteins? Well, there's a couple of reasons for this. Number one, over on the left side here, you can kind of see some things about DNA. DNA is a long-term copy. It's very, very stable. And this is something that the cell needs to preserve and keep secure in the nucleus for basically the entire cell's duration. Not only that cell, but any of the offspring that come from that cell all have to have that identical copy of it. And so it's kind of important to make a temporary copy of it. They use the kind of the CD-ROM version of it here, but they take a version of this DNA and turn it into a copy of it that's more portable, that you can make hundreds of copies of it as opposed to just having one. And then that copy can be actually taken outside of the nucleus to the parts outside of the nucleus, which is actually at the ribosomes, where all of this construction can happen. This DNA stays safe inside the nucleus, locked up, because outside of the nucleus in the cytoplasm, there's actually a lot of really dangerous chemicals out there. A lot of enzymes, a lot of, they call them free radicals, but a lot of free radicals are out there, things that can really break down that DNA and degrade it. So it's kept safe behind this nuclear envelope wall so that it can maintain itself, pretty much intact without really a lot of risk of mutation for generations. The RNA copy, on the other hand, is pretty unstable. Chemically, it doesn't last very long. So it rapidly gets broken down into its component parts pretty darn quickly by enzymes and other just oxygen and other compounds that are outside in the cytoplasm. So this copy doesn't last very long, but it can be used very, very quickly. And it's used out to make, eventually to make proteins in that process called translation. So again, we have to have transcription to keep that DNA solid, to keep it safe, to keep it long-term inside the nucleus. The transcription process makes this portable, disposable copy that can then be used. Not only one copy, but actually hundreds of copies, but then they can also be broken down relatively quickly. So a little bit of how this works. The process of transcription is almost identical to the process of replication. Very, very similar process. And so you can see that the DNA gets unwound, and you can kind of see those two replication forks kind of in here. And then it's complementary base pairing. If you look at here really closely, you've got one side of the DNA, and it's called the antisense strand, which I know is kind of a funny name for it. But there's a big enzyme that comes along that helps kind of read what the DNA strand says, and then it lays down complementary base pairs. So if you look at this very closely, you can see it's pretty much similar to the replication process, that T and A go together, C and G go together, G and C go together. There's one big difference, though, that you have to see, and that's down here. See the letter U? The letter U is a different nitrogen base called uracil that is only found in RNA. It takes the place of a T, thymine, in RNA. So it's a little bit of a different structure. You'll see more about that on the next slide. But in any case, that complementary base pairing that you learned about before, basically the same idea. Enzymes come along and mediate it. complementary base pairs are laid down. The only things that you need to know that are a little bit different is that only one side of the DNA is used, the other side is not. This side, the sense strand, is actually kind of a cap, I guess, is what gives it that double helix shape. But really only one side is read and transcribed, and that we get an RNA transcript. It's a slightly different chemical than DNA is. So let's take a look at how those differences are in the next slide here. So let's take a look at the key differences here between DNA and RNA. Here's your DNA over here on the far right. This is what you're used to seeing. It's got this double helix sort of shape to it. There are two sugar phosphate backbone molecules. Those are like the sides of the ladder. We've got our 4 different nitrogen bases: cytosine, guanine, adenine, and thymine. And then they're bonded together in complementary base pairs across the center to give it that full double helix ladder shape. A couple key differences between DNA and RNA. Number 1. RNA is only a single strand. There's only one of these side backbone pieces. So kind of imagine half a ladder split down the middle. The nitrogen bases in the middle don't form a complement. They stay as only a half of a rung. So you can imagine it would totally not be useful as a ladder, but they do complementary base pairing later, but when they're sitting kind of in their sort of stable format like this, really they don't have the doubled up format like these guys do. It's just a half a ladder. Nitrogen bases are basically the same. 1, 2, 3 of them are the same. There's one key difference, and that uracil is substituted for the base known as thymine. So in DNA we get thymine bonds with adenine, but in RNA we would get uracil instead of thymine, and uracil would match. There's really only a couple of little tiny differences, basically kind of what's going on over on this side, but for whatever reason, uracil does not bond with the RNA sugar molecules like the thymine does in the DNA sugar molecules. So just a quick substitution. The other thing that's different has to do with the name. Why is one called RNA, ribonucleic acid, while the other one's called DNA, deoxyribonucleic acid? And you can see there's this one little tiny difference down here in this ribose molecule. The DNA is deoxygenated in that one spot. RNA has it, and that has an oxygen at that spot. So just a slight difference. This is called ribose. This one's called deoxyribose, hence the names RNA and DNA. And then the last thing that's different between them is that RNA actually, because of its single-stranded nature, has a number of different folded up forms. DNA only has one form. It's always this double helix form. It's always, in either the chromatin or the chromosome form, but it's always this big double helix. RNAs, because of its single-stranded nature, can take on a bunch of different folded structures to it. We're going to take a look at RNA a lot in this particular video, which is just a single strand, kind of like is pictured here. tRNA and rRNA are some different folded up versions of it. This is actually the stuff that makes ribosomes. That's what the little R stands for out in front. The M here stands for messenger. T here stands for transfer, and it's got kind of a clover leaf shape, and there's actually a whole bunch more. So again, those are the key differences. You've got a single-stranded versus double-stranded. You have a slightly different base, uracil versus thymine, and you have a different sugar in that sugar molecule, plus the fact that you've got a bunch of different forms of RNA versus one single form of DNA. So, once we've got the RNA has been made, and we go through that process of complementary base pairing, we get our single-stranded RNA strip, we get something called pre-RNA, and this is what this piece would be up here. It's actually just a direct letter-by-letter copy, complementary copy of what that DNA is. And it turns out, we found this out relatively recently, that this is actually kind of a rough draft, that some editing has to be done before this transcript gets turned into its final mature form. And this is the difference between what you see up in this top thing, exons and introns. It turns out that exons are parts of the RNA that actually get used to make protein eventually. And introns are little sections, and it's sort of odd, or in between, that really don't do anything. And they actually get cut out. It's called excised is the name of that. But these little introns get removed before we get to our final mature RNA. So we start off with a complementary strip like this one that's complementary to the original DNA gene. And then once it gets outside, we do a little bit of trimming. We edit out some of that extra stuff called introns, and we kind of re-glue those pieces back together to get our mature mRNA. And again, the M here stands for messenger. It's carrying the message of that DNA gene out into the cytoplasm. Last couple things get done to it, just to kind of stabilize it a little bit. You can kind of see that same idea of exons and introns here, but a little bit of a cap gets put on the front end, a little bit of a poly-A tail gets put on the back end. These are simply the chemical versions of handles, so that the enzymes that are used in translation, the next step, have some place to sort of grab on and start, and then eventually be able to let go. So again, a little bit of modification is done from that original transcript, which is just that basically letter by letter complementary copy of the DNA, to a kind of a trimmed down edited copy, and then finally a copy that has a cap and a tail to it. Moving along is this next idea of what happens to this transcript once something gets done with it. The one gene, one protein rule is what we call the central dogma of genetics. I know it's kind of a funny name, and here's our central dogmas over here. It's actually a dogma is sort of a rule that cannot be violated, sort of like a code of ethics to some extent. But it used to be thought for quite a long time that if you had DNA, that all of the DNA was transcribed into RNA, and then all of that RNA was transcribed into protein. Basically, one gene would eventually determine one protein through the processes of transcription and translation. However, we started to realize that there were some differences, and this is kind of modern genetics. That number one, there is our funny little backwards dog here. There were some cases where actually reverse transcription could happen, where RNA can get turned back into DNA. It's actually one of the key things that some viruses do, especially the HIV virus. And that was one of the reasons why we started to realize that maybe the central dogma of genetics maybe wasn't quite as hard and fast a rule as we thought. But this typical direction, this typical flow of information from DNA through RNA through protein holds most of the time. So let's take a look at how some of the cases maybe don't necessarily match that rule. So what we're starting to find now in modern genetics is that this idea, and it kind of shows it up in this top left diagram here, this idea of making a transcript that carries that message is pretty much true. But there's some things that we now know about DNA that maybe don't eventually end up in proteins. They're still important. So you can see in this top one here, there's a little region called a promoter and a terminator. These are parts of genes, kind of the on-off switches, that sort of help the RNA enzymes sort of know where to attach, start attaching, and then know when to let go. So these would be parts of a gene, but really don't end up in a protein. Furthermore, this bottom left one here, we now know that certain genes don't end up in proteins at all. Certain genes, their whole job is to make transfer RNAs, or to make ribosomal RNAs. That's this rRNA. And snRNA is small nuclear ribonucleic acids. These guys act like a type of enzyme that, even though they're like enzymes, they really aren't protein at all. They're actually made out of nucleic acids. So we now know that there are other genes that really don't do anything in terms of protein at all. There's only a small number of our genes that actually end up being proteins, and these are the ones that make mRNA. So there's a couple of different variations there. And then finally, another thing that we realize, and this is that idea of introns and exons, is that what an intron versus exon is kind of depends on what protein is being made. And you can kind of see here is one final transcript, here's a second final transcript, actually using different parts of what we originally thought of as being a gene. So when we take this first one to get protein P2 here, whatever that is, or this one to get protein P1, they actually use different sections of a gene. So to get this first one, exon number 1, exon number 3, exon number 4, exon number 5 are all put together to make this protein. Whereas in this one down here, maybe a different sequence of exons. Exon number 2, exon number 4, 6, and 7 get put together to make this one, which eventually results in a different protein. So again, we know that DNA, most of it, codes for proteins. And almost all of it codes for proteins maybe indirectly. But it still isn't as simple as one gene makes one protein. A little bit messier than it used to be. Okay, last one. In terms of if you're looking for a 4, here's some ideas for you. What are really the key things I need you to get out of this? I need you to understand, in this particular unit, the big differences between DNA and RNA. Those structural differences, the different kinds of sugars, the different bases, things like that. I need you to understand the process of transcription is basically complementary base pairing. And then I need you to kind of understand that that transcript undergoes some changes before it gets finally used. If you're looking for some differences, for some fours, maybe something a little bit higher level, you can look a little bit more in depth into some of those things I just skimmed over, like some of those things about how is the central dogma not so central anymore. All these different crazy kinds of RNAs. Here's actually a map that somebody made of some different kinds of RNAs. Somebody has actually put forward a hypothesis that RNA used to be the genetic material of choice instead of DNA. You could take a look at some of those other things that I alluded to, some of this non-coding DNA, what is it used for? Why exactly do we have so many introns, and what are they used for? You can look into some of that process of removing those exons, and there's a structure called a spliceosome, great name, that does that. You can look at the specific enzymes that are involved with this. There's a bunch of enzymes that were involved in replication, where there's just as many that are involved in transcription, and you can do a little quick demo on that. Or you can take a look at the role of reverse transcription and retroviruses and how that really turned the whole central dogma up on its head. In our next video, we're going to take a look at translation, which is that next step, taking that mRNA, that now final copy, and then turning it into something that resembles a protein. There you have it. See you in class tomorrow.